My name is Alex Rajewski, and I'm from the University of California, Riverside, and I'm here to tell you a little bit about some of my dissertation work into the divergence between dry and fleshy fruits. But first, I kind of want to give you a bit of a sales pitch on my study system. So I work in the nightshade family, Solanaceae, and I think it's really great for comparative studies of fruit type. Here's a phylogeny, and if I color code all the genera here by what type of fruit they have, either dry or fleshy, a really interesting pattern emerges. So all of these early diverging taxa down here form a grade that is overwhelmingly dry fruited. There's a couple of exceptions, but the pattern is pretty clear. And then with the divergence of the Solanoideae clade, which is bracketed here, there's a shift in fruit type from an ancestral dry capsule to a more derived fleshy berry. And so the two most prominent examples of this would be tomato, which of course has a fleshy berry, and tobacco, which has a dry base and capsule. And consequently, those are the things that I focus on for the rest of the course. But there are a handful of other important species in this family that don't get nearly as much face time as I think they should. So petunia, eggplant, bell pepper, and then a bunch of other medicinally important taxa. And additionally, the family is really great for doing comparative genomics and genetics and transcriptomics because there are a number of sequenced genomes. So I've highlighted all the genera here that have sequenced genomes. There are a handful of tomato genomes, a handful of tobacco genomes. We're in the process of sequencing Deuterostromonium, but it's a really convenient framework to do a lot of comparative questions, especially between dry and fleshy fruits. And so I know that when I say dry and fleshy fruits, you have like a very intuitive understanding of it. No one's like being blown away right now. But I want to make it a little bit more pointed. So when I talk about fleshy fruits, I'm almost always talking about the berry of tomato, and dry fruits are the capsule of tobacco. And one of the first things you can see when you look at the fruit wall and staining with safranin is that tobacco has this dark band of lignified tissue. And this is very obvious from handling a tobacco fruit. It's very woody. So one of the key differences is lignification of these two fruits. Tomato doesn't lignify, tobacco does. Another thing that's probably maybe visible in these micrographs, but is not very obvious, is the cells themselves. So as you go from the inside of the pericarp to the outside, so from left to right in your perspective, if you count the number of cell layers, tomato has many more than tobacco. Tobacco pretty consistently has 7 to 8 cell layers across species, whereas tomato, depending on the cultivar, has many more, generally in the dozens. So another difference is the number of cell layers, and in tomato, the cells themselves in these layers tend to be larger. And so this is the basis of the size differences, not just number of cells, but also the size of the cells. Importantly, this is just the terminal phenotype, but we know that fruits are very dynamic organs across time. And so here's a developmental series of tobacco up top and tomato on the bottom, from flower to fruit. And you can divide fruit development into 4 stages. The first is a stage of ovary patterning, which is really important for these two species because it's actually very similar between the two. If you take cross-sections of tobacco and tomato ovaries, they look not indistinguishable, but very, very close. And then stage 2 is triggered by fertilization, and this is a burst of cell division that gives the fruit pericarps at least almost the final number of cells they're going to have. Stage 3 is much longer, and this is where the two fruits really begin to diverge. So in the case of tobacco, it's almost always lignification. The fruit's getting woodier and woodier. In tomato, there is a lot of cell expansion and endoreduplication that goes on. And then the final stage is maturation, which in the case of tomato is ripening. So you transition from being a photosynthetic organ to mostly a carbohydrate storage organ. The fruit cell walls soften, and then you get a lot of secondary metabolites that pop up, carotenoids or other flavor compounds. In the case of tobacco, the fruit begins to dry out, and this mechanical tension from drying actually allows the fruit to split open and release its seeds. And so our question is, what genes are expressed in common throughout this developmental series between the species, or which genes are not expressed in common, but very much a comparative study. And so we want to take RNA from both species at each of the 4 stages. And my lab mate, Dinusha, who's not giving a talk unfortunately, has managed to collect this entire series from tomato. I've been working in tobacco and have been having slightly more difficulties. So stage 1 and stage 3, I have tissue from those. Stage 2 is a little more challenging. Stage 2 isn't as discrete as the other stages. And so if you have a cross-section through about a 5-day-old fruit here, stained again with safranin, you can see that the apex is very lignified and is in stage 3, whereas the base isn't and is in stage 2. So if you collect an entire fruit at this day, you're going to get a mixture of tissues, which is rather unfortunate. So I've been doing some in situ hybridization to actually time when this cell division happens, and I figured it out, but it is very tedious to collect as much small tissue to actually get my time point. So this is coming soon. It's been figured out. We're just collecting tissue. For stage 4, the problem is that it's dry and very papery pericarp, so it's very hard to get RNA out of dead tissue. but I'm working on it. I think I've got some tricks up my sleeve. So for the rest of this talk, I want to focus on stage 1 and stage 3 in both the species. And so here is my sort of method slide. I've got all 4 of my tissues. I've collected RNA in triplicate. I'm happy to talk about my RNA analysis package if you'd like, but it's kind of dry. The first set of results I want to present are within species, but across time comparisons. So I'll do tobacco first because it's better, and tomato. Tomato has a pretty well annotated genome, and so I was able to do a Go enrichment very easily. For tobacco, the genome is very much a draft genome, so I assigned Go terms. I'm also happy to talk about that, but the tobacco genome is not nearly as useful for a lot of these functional studies. So in my first comparison, I'm comparing tobacco, stage 1 versus stage 3. And genes that are upregulated at stage 3 are pretty much carbohydrate metabolism, which is kind of a boring Go category, actually. But we do see some things that are more indicative of what's actually going on in this fruit. So we see lignin biosynthetic processes, plant-type secondary cell wall remodeling is going on, cell wall organization. So it's very heartening that our Go terms are at least consistent with what we'd expect to be going on at this fruit, at this stage. And we also see a handful of photosynthesis-related genes, which makes sense. The fruit is beginning to mature, but it's not nearly mature, it's still photosynthetic. The same comparison in tomato overwhelmingly is related to metabolic processes, which again is a very broad Go category, but I think it's important to point out what we're not seeing. We're not seeing any other non-metabolic Go categories except photosynthesis. And if you sum up all of these metabolic categories, you get something like half the genes that are differentially expressed are involved in metabolism. So the tomato fruit is overwhelmingly a carbohydrate metabolic organ. And to really drive home how important that is in this analysis, I want to show you a hierarchy of Go terms. So they are hierarchical and they're interrelated. And you're not meant to actually read the terms, but here is the biological process Go category, which is the most broad you can have in this group. They're colored by significance, and then once they pass a threshold, they become boxes. This is the metabolic process. So it's the second most broad Go category you can have, and all the other ones are subordinate to that. So it very, very overwhelmingly a carbohydrate metabolic organ. And this is all interesting, but the title of my talk is Conserved Regulatory Modules. So how do I go about finding conserved genes? Tomato and tobacco don't have a common transcriptome, so what do I align my RNA-seq data to? So what I did was, I first looked at my data and asked, does a gene in either genome have a one-to-one ortholog between species? And so there's a bunch of ways to do that. I decided to use a best reciprocal hit method. I used diamond instead of glass because it's faster, and there are some drawbacks to that, but I'm happy to discuss those. And then, given that a gene has a one-to-one ortholog, I asked, is that gene actually present in my 2 RNA-seq data sets? So in the case of the early stage comparison, tobacco has about 800 upregulated genes, tomatoes got about 3,000. And if I look for one-to-one orthologs and then find out which set is present in both data sets, there are about 180 genes. So I lose quite a number of genes doing this comparison. And I think that's going to affect my results overall. Looking at later stages, they've got a more comparable number of differentially expressed genes, just over 2,000, and so I find many more common genes. So I've got about 300 there in this analysis. And when I actually do the Go enrichment, so this is going to compare stage 1 tomato and stage 1 tobacco, looking at genes that are upregulated at this stage. It's a lot of housekeeping, cell cycling, signaling sorts of things. So about 10% of my genes are involved in regulation of transcription. And these graphs are, the x-axis here, although it's technically rotated, is essentially the p-value, and then they're color-coded by how many genes fall into this category. So a lot of genes fall into the regulation of transcription category. But we also see genes involved in signaling, other transcription, and then more broad ones like multicellular organism development, which is, like Alan said, kind of hand-wavy. And when we look at later stages, so this is again comparing stage 3 tobacco with stage 3 tomato, we see a lot of metabolic processes again, probably driven by the fact that tomato is overwhelmingly metabolic. But this suggests that tobacco is not metabolizing carbohydrates. So this is interesting, but we also see a handful of other ones that are involved in cell wall modification, lignification, secondary cell wall sorts of processes, and then a lot of glucose metabolism. Again, I think probably driven mostly by tomato. So overall, to conclude, the early genes in both species are very much related to transcription, signaling, cell cycling sorts of things. The late stage genes are involved in cell wall remodeling and carbohydrate metabolism. And this is pretty consistent with what we see in just looking at the gross morphology of the two species. Where they begin to diverge is that tomato has metabolic genes that are related to glucose biosynthesis or different flavor compounds. So fatty acids or oxoacids or malate, other things that you're going to taste in the final fruit, ideally. And then tobacco has many genes related to secondary cell wall synthesis. So again, tobacco is going to lignify, and this is pretty consistent. So in the future, stay tuned for a complete developmental series from tobacco and also my Datura genome project at Next Botany. And so with that, I want to say that it takes a village to raise a graduate student. And so I want to thank my major professor, Amy, who loves karaoke, and she's so engaged right now. It's so beautiful.
That wasn't karaoke, that was just the Wednesday evening.
It's just a typical Wednesday.
Yeah, but
I was trying to give you help there, but and then Dinusha, who generated the tomato data. I've had a couple of phenomenal undergrads who've been with me throughout this project, and then funding by a lot of grants from UCR and the NSF. And so with that, if you have any questions, I would be happy to take them.